Now let's have a look at how saliva is secreted, which we said in the last video was one of the important secretions of the GI tract, which helps break down food. So salivation is controlled by the autonomic nervous system and is mediated by the anticipatory cephalic phase of digestion. Aldosterone also promotes ion exchange in the salivary ducts. So you have two groups of salivary glands, the sublingual or submandibular glands, which produce a serous and mixed mucus secretion, and the parietal glands, which just produce a serous secretion. So if we zoom in onto a salivary gland, this is a set of cells which forms a intercalated duct, and striated duct, and an acinus. So the acinus cells are what secretes enzymes, proteins, into a primary secretion here in the lumen. And myoepithelial cell contraction empties the saliva into the ducts. So firstly we have the acinar cells secreting this primary secretion, which is high in sodium chloride, and it's isotonic to plasma, so it draws water into the lumen as well, which increases the water pressure and pushes the primary secretion into the ducts, as well as, of course, the cell contraction. So after it's pushed into the ducts, it gets modified, and this makes it more hypotonic, so it increases the bicarbonate and potassium concentration in the lumen and draws some of the salt, the sodium chloride, out. This is how the composition changes. So this is flow rate. When we're fasting, we're not producing too much saliva, so it goes through very slowly, and you can see that there's a low concentration of bicarbonate and sodium ions, and then a bit of a higher concentration of potassium and a little bit of chloride as well. Now, when there's high flow, for example through a meal, the ducts don't have much time to modify it. Bicarbonate remains high, sodium chloride remains high in the saliva, so it's closer to what the primary secretion looks like. So let's have a look at the secretion in more detail. So these are the acinar cells, which secrete the primary secretion into the lumen. So we have a ATP pump, a sodium potassium ATP pump, which generates an ion gradient. So it generates a more negative gradient inside the cell and generates energy. So it causes 3 sodium ions to move out and 2 potassium ions to move in. This powers an NKCC, or a sodium potassium chloride transporter, and this drags in sodium, 2 chlorides and a potassium ion. The potassium exits through a channel and the sodium exits through the ATP pump. The chloride is then dragged through the cell and into a CFTR, or a luminal membrane transporter, which is a channel which allows the chloride ions to go through to the lumen and increase the chloride concentration, and water and sodium ions are also transported paracellularly down their concentration gradient. So it has a high concentration of sodium chloride in the primary secretion. The duct cells, so as the primary secretion passes through the duct cells, it's modified like this. So the lumen, there's another one of these ATPases which cause a gradient to be formed. Then now we have more transporters. So we have a sodium hydrogen transporter and a hydrogen ion and potassium transporter, and the hydrogen ions are cycling back and forth between these two transporters, whereas this is causing potassium to go out and sodium to be absorbed back in and into the blood, and sodium also goes through channels as well. And then we have a chloride bicarbonate transporter, which switches bicarbonate for chloride ions, and then the chloride ions exit through this channel here. So the net effect of this is increasing the amount of potassium and increasing the amount of bicarbonate in the saliva. Why do we need to do this? Well, some people say that increasing the potassium, exchanging potassium for sodium, makes us taste sodium better, and we need sodium to survive, so it's an evolutionary trait maybe. Whereas increasing the bicarbonate levels in the lumen is helpful because it buffers the saliva, and it can act as a buffer if we're, for example, vomiting and acid gets into our mouth. Now the functions of saliva, we have 6 main functions here. So firstly, lubrication. There's mucin glycoproteins contained in the mucus of the saliva, and secreting glands for the solution of food, swallowing and speaking. Defense. The saliva contains antibodies and immunoglobulins, lactoferrins which bind iron and depletes it for bacteria, protein-rich proteins which neutralize plant tannins which are toxic, and lysozyme antimicrobial enzymes. Buffering, so bicarbonate ions buffer the pH of saliva and buffer acid that might come into the mouth. Thermoregulation, digestion, so salivary amylase digests starch into oligosaccharides and digests around 75% of the starch in a meal. Continues activity for around 30 minutes when protected in the food bolus, and it protects against stomach acidity, which is similar to buffering. So vitamin B12 binds haptocorin secreted in the saliva, which protects it against the acidity in the stomach, and B12 is an essential nutrient. So the PNS and the SNS, the parasympathetic and sympathetic nervous system stimulation, can increase salivation. So both of these can have the same effect on the salivation, and this is how. So the PNS secretes acetylcholine and vasoactive intestinal peptide, which causes vasodilation, metabolism, and growth. So there's two ways they can do this. So the sympathetic system releases noradrenaline from nerve terminals, acts on the B1 adrenergic receptors, and increases levels of cyclic AMP, which causes increased exocytosis and increases enzyme release from the glands, the duct cells or the acinar cells in the salivary glands. The parasympathetic nervous system increases myoepithelial cell contraction, opens more acinar cell channels, and it does this by releasing acetylcholine from nerve terminals, which acts on M1 and M3 cholinergic receptors, increases the amount of calcium in the cells, and increases channel activity. Now, after the food has been processed in the mouth by chewing and saliva, we need to swallow it. So this is where the bolus is pushed to the back of the mouth. How this happens is touch receptors in the pharynx are stimulated, so this initiates a deglutition reflex coordinated in the swallowing center of the brain, so the medulla and the lower pons. And then there's deglutition apnea, which is the respiratory center of the medulla being inhibited by the swallowing center, and the fine control of striated muscles in the pharyngeal region, which contract and cause the food bolus to be swallowed. After it goes into the pharynx and then down the esophagus, this is what happens. So the esophagus is a long tube leading from the pharynx, back of the mouth, to the opening of the stomach. And the electrical activity, it looks a little something like this. So if we plot pressure against time, we have a primary peristaltic wave beginning below the upper esophageal sphincter, which sweeps the bolus at 3 to 5 centimeters per second. So it's a pretty weak little peristaltic wave sweeping the bolus down. If a bolus is stuck, then persistent esophagus distension causes a secondary peristaltic wave via a local and vagovagal reflex. In the upper third, we have an increased pressure. The middle third has increased pressure, which is more delayed, and then so on in the lower third, until we reach the lower esophageal sphincter. So this has a feed-forward reflex. The sphincter relaxes before food arrives because nitric oxide has been secreted by cells nearby, and this relaxation allows the food to pass through. So this is the upper esophageal sphincter. It has striated cricopharyngeus muscle. The upper third of the esophagus has striated longitudinal and circular muscle. It's innervated by the vagus nerve. The middle third has striated and smooth muscle, so it has a mix, and the lower third only has smooth muscle. The lower esophageal sphincter is important for reducing the gastroesophageal reflux. It has smooth muscle, circular smooth muscle, which is specialized, controlled by the enteric nervous system fibers, with input from the autonomic nervous system. And when the lower esophageal sphincter doesn't work, you can get heartburn and Barrett's esophagus, which are conditions where the acid enters back through the esophagus and causes erosion here, which is pretty painful. This is normally prevented by the lower esophageal sphincter and the cross of the diaphragm. The esophagus secretes mucus and lubricates food to protect the lower end mucosa against acid reflux as well. Achalasia, difficulty swallowing when the lower esophageal sphincter relaxation reflex is compromised, so it doesn't relax before the food arrives here and it contracts, so we can't really swallow very well. And it can be caused by enteric nervous system damage, which makes it not relax properly. Food builds up in the esophagus and can cause the esophagus to distend, causing megaesophagus, which is a condition that needs to be treated. So now we look at vomiting, or empsis, which is where food comes back up from the stomach and back up through the mouth. So it can be triggered by many things. It can be triggered by stretch, which acts on the GI tract, and emetic drugs such as ipecac, which acts on the GI tract and the chemoreceptor trigger zone in the area postrema of the brain. Now this is on the floor of the brain in the fourth ventricle outside the blood-brain barrier, so there's not much protection for it and it's triggered pretty easily by drugs in the blood. So other things which can trigger vomiting, touching the back of the throat, dizziness, genitourinary pain, and psychological upset. This all acts on the vomiting centre in the brain, the medulla, and the GI tract and the chemoreceptor trigger zone also act on the medulla vomiting centre by the vagus nerve, and this all causes vomiting. There's 5 things which happen when you vomit. Firstly, the lower esophageal sphincter relaxes and you increase salivation to help buffer for the contents that are potentially coming out. Number 2, there's retroperistalsis. So this is peristalsis not in the anal direction, but in the oral direction, in the opposite direction. Waves of contraction from the middle of the small intestine propel to the stomach and through the relaxed pyloric sphincter. Thirdly, there's decreased intrathoracic pressure if you inspire against a closed glottis. So it's like the reverse of the Valsalva maneuver. Number 4, there's increased abdominal pressure. So abdominal muscles contract. And number 5, there's retching. So if the upper esophageal sphincter is contracted, then the vomit doesn't come out, but it can go back into the stomach. And this can happen several times before you actually vomit it out. In severe vomiting, bile can enter the vomit because of duodenal contraction. Rabbits, mice and rats can't vomit, which is an interesting fact. Now we move on to the stomach. So there's 5 main functions of the stomach. It's a food reservoir. It facilitates digestion, so it initiates protein digestion because there's proteases in it. It destroys ingested microbes because it contains acid around pH 1, pH 3, and it helps regulate appetite, so it feeds back to the brain on how stretched it is. It regulates later gut activity as well, so there's feed-forward systems. For example, esophageal stretch can cause receptive relaxation of the stomach. The fundus and the body of the stomach relax via the vagovagal reflex, and around 1.5 litres of food can be accommodated in the stomach, so quite a lot. This is the stomach, and it can control emptying rate. So firstly we have the esophagus entering the stomach through the cardia, the fundus, the body, the antrum, the pylorus, and the duodenum. So the cardiac glands in the cardia secrete mucus, and the fundus has no slow waves really. The slow waves begin at the body. So liquid contents can enter the duodenum, but much tougher food remains in the stomach until it's digested. If we look at this graph, plotting time in hours against percent emptied, someone put radio-labelled glucose solution. So if it's in solution, then it's emptied pretty fast, but if it's solid, then it is stuck in the stomach and has to be digested. So there's a lag phase with radio-labelled diced liver, and someone put radio-opaque plastic spheres, 7mm plastic spheres in them. The emptying is not going to be very good there, but you get the idea. So in blue we have the secretions of stomach, and in green we have some bits about the electrical activity. So the body has oxyntic glands which secrete hydrochloric acid, some enzymes like pepsinogens, chymosin, and other enzymes, intrinsic factor which helps B12 from the pancreas in dogs and cats, but in the body of the stomach in humans, and mucus as well. So the body electrical activity looks something like this. And so we're plotting time against depolarization, and it depolarizes first. So the pacemaker zone has interstitial cells of Karl, which generate slow waves around 3 per minute. They propagate down and stop at the pyloric sphincter, so the pyloric sphincter lacks interstitial cells of Karl, and action potentials, little action potentials, are superimposed on the plateau. Gastric smooth muscle contraction depends on neurons and hormones, and in the fed state, increased acetylcholine and gastrin causes increased plateau duration. So we have a plateau here of the slow wave, and if we're fed, then more acetylcholine and more gastrin will be produced, which increases the plateau. Now in the antrum, there's much thicker muscular walls which increase contraction force, so you can see that there's a lot more action potentials and a much longer plateau here. In the pylorus, there's circular muscle, and there's pyloric sphincter tone control. So the pyloric sphincter is usually narrowly open and relaxes between contractions, which allows food into the duodenum. The enteric nervous system has inhibitory fibers, which secrete nitric oxide and allows it to relax. Retropulsion is where the contents are forced back up as the pyloric sphincter contracts, and the antral mill, which is a bit like segmentation in the stomach, it squeezes back and forth and breaks down large particles. It mixes the food with gastric secretion to form chyme. In the last part, the duodenum, before it goes to the intestine, well, the duodenum is part of the intestine, but anyway, the duodenum has nutrient sampling. There's feedback on the rate of emptying. For example, acid, fat, and peptides appearing in the duodenum in the small intestine too fast slows down the rate of emptying. The pyloric glands secrete mucus and gastrin into the blood as well. Enterogastric reflex is when the distal gut, for example the pyloric sphincter, is stretched, and neuronal and hormonal reflexes slow the gastric emptying. So the nutrient sampling is contributing to the enterogastric reflex. The migrating myoelectric complex is where contractions sweep the remaining food down into the duodenum. So remember here, some food, such as these plastic spheres, won't be emptied very fast, so we need something to sweep them down. And MMCs are waves of contractile activity to the terminal ileum, which is initiated by the hormone motilin. And you can see that in different parts of the stomach and small intestine, there are slow waves happening at different times. So at 6 p.m. you might have a wave which is happening in the antrum, and then a little later, 6:30 maybe, you might have one in the duodenum, and then in the jejunum, and passing the food down into the ileum. So stomach secretions, there's hydrochloric acid obviously, and there's also potassium and sodium which we need to consider. So in the fasting state, there's not much potassium, there's more sodium and not much HCl being produced, but when it's high in a meal, there's a lot more acid being produced here. The oxyntic or parietal cells secrete hydrochloric acid and intrinsic factor. Hydrochloric acid has many functions. So first, it catalyzes cleavage of inactive pepsinogens into pepsins and prochymosin into chymosin, which curdles milk in non-human neonates, but humans use pepsin. And the breakdown of these inactive enzymes into active enzymes is pretty important. It also provides a low pH environment, an acidic environment less than pH 3 for pepsins to work properly. It delays gastric emptying, increases the calcium absorption and solubility, and iron as well, and it destroys microbes and helps release vitamin B12 from food. Intrinsic factor is the only secretion which is essential for life because it binds B12, which is very important. And it's a glycoprotein. It's released in the small intestine. It resists protease digestion and is endocytosed into the ileum epithelial cells. The chief or peptic cells secrete pepsinogens and prochymosyns, which are then broken down. Chief cells secrete these enzymes in response to acidity through the vagal acetylcholine and the cholinergic reflex. The pyloric glands in the pylorus secrete mucus from mucus-secreting cells, gastrin from G cells into the blood, not the lumen, and gastric lipase enzyme as well. The parietal cells can secrete hydrochloric acid, as we saw, and this is called an alkaline tide. So the carbon dioxide is removed from the plasma, bicarbonate is added, and this increases the pH of venous blood. So the parietal cells secrete HCl and add bicarbonate into the blood, and this makes the blood a lot more alkaline. And the way this happens is second messengers. So CAMP, cyclic AMP, and calcium increase the number and activity of transporters. We'll have a look at how that happens later. So this is the lumen, and this is the extracellular fluid, which goes into the blood. So we have our ATPase pump, as always, 3 sodiums out and 2 potassiums in. Then the potassium goes out in both directions through channels. There's an ATPase pump on the luminal side as well, which trades the hydrogen ions for potassium ions. And these hydrogen ions come from carbon dioxide and water inside the parietal cell, reacting to form a hydrogen ion and bicarbonate, catalyzed by carbonic anhydrase. And the hydrogen ions go out on the luminal side, meanwhile the bicarbonate ions go out on the extracellular fluid side, and are traded for chloride ions, which then exit with the hydrogen ions into the lumen. So we have increased concentration of acid here. Things that can increase and decrease this, so we have acetylcholine can increase this by many ways, by stimulating all of these cells, which also increase it. And it's released from nerve terminals of the parasympathetic nervous system. So G cells ECL cells, enterochromaffin-like cells, and D cells. So the G cells, they work via local stretch reflex, and they are vaguely stimulated via GRP peptides in the stomach lumen, the antrum, and the duodenum. So G cells secrete gastrin into the blood, and the action of secretin decreases the G cell activity. But anyway, G cells secrete gastrin into the blood, and gastrin increases the activity of ECL cells, as well as increasing the calcium concentration in the parietal cells. Increases in calcium concentration, which is also potentiated by acetylcholine, increases parietal cell HCl secretion. Now, ECL cells, or enterochromaffin-like cells, are in the gastric glands, and they secrete histamine, and histamine is the strongest agonist of HCl secretion. Histamine acts on H2 receptors in the parietal cells and increase the cyclic AMP levels, which also leads to parietal cell acid secretion. D cell secretes somatostatin, which decreases the level of cyclic AMP, decreases the parietal cell HCl secretion as a result. Now there's also negative feedback, so when there's a lot of acid detected in the lumen, the D cells detect this and increase their activity, so it decreases the acid secretion to protect the lining of the stomach. S cells release secretin, which decrease the HCl secretion through vagal afferent fibers and also decrease the activity of G cells. And prostaglandins decrease the HCl secretion, as well as increasing bicarbonate and mucus secretion, so it protects against acid. Now, there's phases of acid secretion. Firstly, before eating the meal, there's the cephalic phase, and this accounts for about 30% of the total secretion. So this is a feed-forward signal, where there's sight, smell, and taste of food causes acetylcholine to be released from nerve terminals in the parasympathetic nerve system and increase the HCl secretion via all these methods. But there's usually negative feedback through the somatostatin, so there's not much of a pH change. The second phase is the gastric phase. So this is the response to food in the stomach, and stretch causes vagovagal and local reflexes and causes increased acid secretion. The H plus is buffered by proteins in the food, and proteins increase gastric secretion and increase the hydrochloric acid concentration. The last phase is the intestinal phase, which is where most of the secretion happens. So this is what happens when chyme enters the duodenum, and increased stretch causes a vagovagal reflex again. The duodenal G cells secrete gastrin and decrease the hydrochloric acid secretion later because of the acidic duodenal contents. So we can plot some charts here of when stomach volume increases. So this is time against stomach volume. So if stomach volume suddenly increases, then the HCl rate will gradually increase following that, and the luminal pH will increase sharply as well. Acid in the duodenum causes increased secretin release, and secretin does two things. It increases the bicarbonate secretion by the pancreatic duct cells and decreases the acid secretion in the stomach, all leading to decreased acid in the duodenum. Now, GORD and stomach ulcers. So GORD stands for gastroesophageal reflux disease, and stomach ulcers can also be caused by acid. So stomach mucosa is protected by gastric epithelial cells, mucus and bicarbonate containing, which keeps the stomach within pH 7, around within 200 micrometers of cells, despite it being a very low pH of 1 or 2 in the lumen. NSAIDs, or non-steroidal anti-inflammatory drugs, for example, aspirin and gram-negative helicobacteria, inflame the stomach wall and predispose people to ulcers by destroying epithelial cells, and this can lead to Zollinger-Ellison syndrome, which is a gastrin-secreting tumour, which, as you know, gastrin causes increased acid secretion, so it can cause GORD. Stress, alcohol, and smoking are risk factors, and helicobacteria also burrows into the safe pH 7 mucus layer, so you can't really kill it with the acid. Prevalence is significantly increased in developing countries as well. Now, the pancreas. So the pancreas has many functions. It has exocrine and endocrine functions. We're going to look at the exocrine function here. So it secretes many enzymes, and it secretes bicarbonate as well. So firstly, the pancreatic duct secretes bicarbonate-rich pancreatic juice. So this is the lumen and this is the ECF. This is a pancreatic cell. Carbon dioxide and water, coming from acid and bicarbonate, join to form bicarbonate and acid again, carbonic anhydrase. Then the acid goes out and the sodium comes in and goes back out again through the ATPase, which causes a gradient to be formed. and potassium comes in. Potassium goes back out again, and sodium is dragged through paracellularly into the lumen. But the main thing here is that chloride is switched for bicarbonate, which is produced from this reaction. And chloride goes out, bicarbonate goes out, and sodium goes out into the lumen. Now, how is this controlled? So there's sympathetic and parasympathetic control. Sympathetic control involves vasoactive intestinal peptide, which acts on its receptor and increases cyclic AMP levels in the cell. Secretin also increases cyclic AMP levels in the cell and increases the CFTR, the chloride membrane channel, which causes more Cl- to be secreted into the lumen. Parasympathetic can decrease the amount of bicarbonate and chloride which is secreted, and it does this through acetylcholine, so acting on M3 receptors to increase the amount of calcium in the cell, and then increases the amount of potassium that is secreted into the extracellular fluid. And cholecystokinin can do this as well. So if there's a great increase of chloride secretion, then water will be dragged through following it. And cholera is a disease caused by drinking dirty water, so bacteria, which can increase the cyclic AMP levels, so here, and increase the chloride secretion. Water follows and can cause extreme diarrhea, so people dry out, and that is how they die. It's a very nasty disease. So we've looked at how it secretes pancreatic juice. It also secretes these enzymes, so amylase, lipase, and ribo or deoxyribonucleases. It secretes proteases in the form of inactive zymogens, which need to be cleaved, and PSTI, or pancreatic secretory trypsin inhibitor, which protects the CNR cells from inappropriate trypsin activation.